In this video, we will discuss about how this lipid-rich environment in the SETD2-deficient pancreatic cancer cell occurs, or what is the main cause of this lipid-rich stroma in this SETD2-deficient pancreatic cancer cells. So, in order to find out the main cause of this lipid-rich stroma, they again did the cluster of this cancer-associated fibroblast. As in the previous results, they have found out that cancer-associated fibroblast and the lipid-rich markers are coexist. So what they did is they recluster this CAF of the single cell RNA-seq data into 4 different clusters, and when they check these clusters in KC and KSC derived tumors, they find out that in the KC the C3 clusters are more predominantly exist compared to the C3 clusters in KSC, whereas for the C2 the C2 clusters exist more predominantly in the KSC compared to the KC. And these things they have quantified in the panel B also, where we can see that in the KSC the C2 clusters are exist more compared to the KC. And when they check the differentiation pattern or differentiation pathways, they find out that C2 and C3 have complete different differentiation pathways, and some of the markers that are expressed or overexpressed in C2 are ABC8A, CXCL, 14LPL, whereas some of the markers that are overexpressed in C3 are MFAP4, COL1A1, COL1A2. And after that they check whether these clusters belongs to have this MyCAF signature, ICAF signature, or APCAF signature. And when they check these clusters and their scoring, they find out is that CT clusters is more closely or has a signature. C3 cluster has a signature towards this MyCAF, high signature towards this MyCAF, whereas the C1 cluster shows a signature towards this ICAF, and and C2 clusters has completely different characteristics that does not match up either with MyCAF, ICAF, or APCAF. APCAF signatures are also more closely in line with this cluster C3. When they do the gene set enrichment analysis based on the genes that are upregulated in these clusters, they find out that in the C2 cluster, the ABC transporter or adipose genosis are highly upregulated, whereas in the case of C3 clusters, the oxidative phosphorylations, hypoxia, and collagen fibrils are this downregulated. So all this indicates that in the C2 this adipogenesis and ABC transporters are related to lipid-rich pathways, and also these oxidative phosphorylations are lower in the C3 compared to the C2. This all indicates that the C2 cluster might be related towards this lipid-rich stroma. And when they check the other factors or other genes that are when they check the relative expression of other genes in different clusters, what they have found out is that this PDPN is overexpressed in this cluster 3, ACTA2 is overexpressed in cluster more overexpressed in cluster 3, whereas FAP is overexpressed in cluster 2, and ABCA8A is overexpressed in cluster 2. And thereafter, when they check the immunostaining of the mice models of different cancer types such as this KC and KSC, and what they have find out is that this FAP is more predominantly expressed in this KC type tumors, whereas this alpha SMA is expressed in the KC and normal wild type KC, and the KPC tumors, whereas it is less expressed in the KSC tumors. So this and also when they check about this FAP that is expressed in the KSC, they find out that its expression is closely in line with the pancreatic tumor cells, that is PDPN. So this indicates that the FAP might be the marker for the lipid-rich trauma around the pancreatic tumor cells, and FAP is also overexpressed by the C2 cluster. So this C2 cluster cancer-associated fibroblast are the one that are lipid-rich and that cause this and that will that cause the OXPHOS in the pancreatic tumor cells, and that that area we have covered in the previous videos. And this is the another immunostaining images or zoom in immunostaining images, not zoom in. This is the another immunostaining images. In this also we can see the similar results, that is for this FAP are expressed more predominantly in the case of these KSC tumors, whereas they are expressed less in the KC tumors. In this the bottom rows we can in the bottom rows we can see that difference, whereas in the top rows we can see that the KC and this FAP is can be seen in the KC also and in the KPC also, but in the bottom panel we can see that the FAP are expressed more predominantly in the KSC samples, and they are also closely in line with the PDPN that can be seen here, and this PDPN and also this CK19, which is the tumor cells markers. So this indicates that the FAP could be the marker for the lipid-rich trauma, and after that as this FAP is expressed by the C2 cluster, and C2 cluster has the genes that are upregulated that results in high adipogenesis pathways ABC transporter, and these are the indications of lipid-rich environment. And thereafter they check this FABP plus and ABCA8A plus cells, as we know that C3 C2 clusters has this upregulation of this ABCA8A and also this FAP. So consider these two markers, they check the upregulations, they check its populations in KC and KSC tumor cells, and they find out that KSC cells has more of this FABP plus and ABCA8A plus populations, and these things are quantified on the right hand side of the panel I, where we can see that ABCA8A plus FAB plus CAF cancer, these clusters of cancer associated fibroblasts are higher in KSC cells, and these things they have validated using the body pie imaging, and where we can see that KSC, KFC, and this ABCA8A minus FABP minus cells and the ABCA8A plus FABP plus cells are highly expressed in this KSC cancer associated fibroblast, and the other things that are rich in lipid, or that is the markers for the high lipid stroma, are also overexpressed. For example, this triglyceride are overexpressed in this ABC8A plus and FABP plus cells, whereas the control case ABCA8A minus FABP minus does not have this triglyceride or any FAs, and this right hand side of the panel J indicates the quantification of this fluorescence image, which shows that the body pi MFI is high in the case of ABCA8A plus FABP plus cells compared to the ABCA8 minus FABP minus cells, and this also further substantiate their point that the lipid-rich stroma could be the result of this C2 cancer-associated fibroblast cluster, which are rich in genes that cause this adipogenesis, ABC transporter, and also has this marker FAP markers that later on results in activated OXPHOS or enhanced OXPHOS in the pancreatic tumor cells. So the I hope you understood the main reason behind this lipid-rich trauma in SETD2-deficient pancreatic cancer cells. If you need to understand more about this topic or this lipid-rich trauma in this SETD2-deficient pancreatic cancer cells in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.